Okay, well, I think we can get started. I'd like to welcome the remote audience today, and thank you for joining the Canty Ontario Stage International Speaker Seminar Series. My name is Shelley Bull. I'm an investigator at the Lunenfeld Tannenbaum Research Institute, and I'm also a co-director of the STAGE training program. So I'd like to begin by acknowledging the land on which the University of Toronto operates. For thousands of years, it's been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still home to many Indigenous people from across Turtle Island, and we're grateful to have the opportunity to work on this land. I would also like to mention that STAGE operates with the support of Canty Ontario, which is an extra departmental unit in the Faculty of Arts and Science at the University of Toronto. And the seminars, the international speaker seminars, are sponsored by the Hospital for Sick Children, the Lunenfeld Tannenbaum Research Institute, the McLaughlin Center, and the University of Toronto Department of Statistical Sciences. So I'm very pleased to introduce our speaker today. Professor Wojcik is professor in the Department of Epidemiology at Johns Hopkins School of Public Health in Baltimore. She joined the department in 2019, following time as a postdoctoral research scholar in the Department of Biomedical Data Science at Stanford University School of Medicine. And she completed her doctoral studies in genetic epidemiology at Johns Hopkins. She's involved in leadership of several consortium working groups, including PRIMED, which stands for Polygenic Risk Methods in Diverse Populations, and PAGE, which stands for Population Architecture Using Genomics and Epidemiology, and I think we'll hear a bit more about that today. In 2021, she received the Genomic Innovators Award from the U.S. National Human Genome Research Institute, and she's currently a principal investigator funded by NHGRI, examining the influence of genetic ancestry and population-specific epidemiology on the transferability of genomic findings to diverse and admixed populations. The title of her talk today is Global Diversity, Local Contexts, an Epidemiologic Lens to Modeling Ancestry and Environment for Genetic Risks. And we're really looking forward to hearing on that topic, and welcome.
Thank you so much. Thanks for the great introduction. I will share my screen now so everyone can see it. Okay. All right. Let's get started. And so I encourage anyone, if you have any questions, you can interrupt me, throughout. I think it's probably better to address things as they come up. But yeah, I'm happy to talk about anything that I mentioned today. So as said, my talk today is focused on sort of an epidemiological lens, which is sort of my background and home base is in epidemiology, for the way that we think about how genetics and ancestry might affect risk of different health outcomes. And so we're going to start looking at sort of, as a geneticist, for a long time, myself included, sort of like to think that everything was as simple as, genetics to trait, right? Like that you have a simple thing of looking at the genetics and it leads to the trait, and the world is a very nice place to work in if that's sort of the framework we're working in. That's sort of the way that a lot of studies were conducted for a long amount of time. But if we think about it, that's not really what is happening. What is happening is that there's a lot of different factors that are influencing the genetics that we're capturing, that we're measuring, and how those genetics might relate to a trait of interest. And those include sort of more of a population genetics phenomenon when we think about allele frequencies and LD, also biomarkers and individual level risk factors, but also we can sort of zoom out and think more about how we consider the sort of societal context of the individual, right? So how they identify in terms of racial and ethnic background, the socio-political context, and then sort of broader at the social and structural determinants of health. And so when we go back to that original question of sort of how do we test the association between genetics and trait, and where is it associated, I think it's important to take an even further step back and say, among whom are we looking at, right? Who are we thinking about that? And historically, what we see is that a lot of times these parts of the puzzle, right, these parts of the framework are flattened in terms of you sort of assume a certain level of homogeneity, and that's sort of assumed as part of assumptions of the frameworks, of the methodologies, as well as how we conceptualize our populations. And so what I'm really interested in, my project's really interested in, my program's really interested in, is sort of how do we appropriately account for these factors in a way that both helps understand, the relative role of genetics in human health, but also what sort of part of the human genome also affects the human health within these contexts. And I'm, primarily motivated by this idea of looking at health inequities, right? So we know there are existing genomic health inequities when it comes to technologies that are available to diverse groups. And then even further, how well those technologies do, what that happens further downstream. And it's important to sort of examine for ourselves, sort of how do we get here, right? And sort of why do we keep on making the same mistakes? It's been widely known for a long time that we have a diversity problem in human genetics. But we keep on sort of, recapitulating the same sort of frameworks over and over again, and they keep on coming up. And so if we look at further back and sort of examine how we got here, it's turtles all the way down, right? And by turtles, what I mean is a sort of Eurocentric bias of what we accept as a default. So if we start with the genomic inequities, think about access to healthcare, poor performing tools, and we do a step back into the translational space, we see that often you rely on these big biobanks, and historically those biobanks have been of more European ancestries. And sometimes they aren't even reported as to what the backgrounds of the individuals are. They're sort of assumed to be this sort of nebulous white background for a lot of this work. If you move a step backwards beyond that, a lot of the traditional out-of-the-box methods are considered for single ancestry, relatively homogeneous populations that sort of have to meet the assumption of normality in terms of what we're looking at. This has changed over the past several years in terms of more methods being available, but still often a lot of the methods still rely on some sort of stratification and some sort of level of assumptions of homogeneity. And then we take a step further back from that, we think about who is recruited and how they are measured. Again, historically, we had arrays that were designed just for European ancestries and did very badly in other groups. Newer methods and newer platforms have changed this somewhat, but it's still somewhat an issue. And then also our recruitment is not doing so well, right, in terms of who we include in our studies. And so it's sort of this idea of what do we accept as the default and who has to wait until later on. And so we can see this in the data. So what I'm showing you here is the average sample size for published genome-wide association studies. These are large-scale studies looking for the association of individual markers with a trait. And so what you see here, often you'd see a picture of this looking at the participants, sort of the total number of participants over time. And I think this is a complementary way of looking at it because what you're looking at here is sort of the rate limiting step, right? The statistical power of a given publication and a given study. And what you can see is that over the past, this is from 2012 to 2022, for a decade period of time, you have this sort of astronomical rise in groups that are categorized in the NHGRI, EBI, GWAS catalog as being of European ancestries. And in the dashed line, you have the annual estimate, and then the cumulative knowledge base is shown in the solid line. And this is primarily driven by the availability of UK biobank data, other large-scale biobank data. But what we can see is if we zoom in to just the past, the 5-year period here, where you would have this really astronomical rise in the European ancestries, you don't see the same thing in other groups. You see a little bit of a blip for some groups, and a lot of the groups you sort of see a stagnation where things didn't really change for those groups over that period of time, right? So we still have a knowledge base that is not as diverse as it could be, given the populations of interest when we think about these health outcomes. And so I'm primarily interested in polygenic risk scores, which is sort of built off the back of these sort of GWAS studies and looks to summarize genetic risk across the entire genome. And so, of course, because polygenic scores are built off of often GWAS, you have the same problems being amplified, right? And so here what I'm showing you are the published polygenic scores as of last year. This is 2023. On the left-hand side, I'm showing you, proportionally speaking, each one of these blocks is not meant to be a score, but rather proportional to the entire knowledge base. How many of them have only individuals of European ancestries? That's sort of the dark blue color. And then those that have European ancestries, and they've also included other groups. And you can see that there's some that have a more diverse representation. Now, my research primarily focuses on populations in the United States, primarily minoritized groups that are recently admixed, so African American, Afro-Caribbean, Hispanic, Latino groups. And what you can see here is just a small proportion of those include anybody who ever identifies as anything that would then fall into those categories, right? And so given that, I work in the United States in that context, it's important to think about how that works in terms of our actual populations. And what you can see on the right-hand side for the 2020 US Census is that, it's not reflective of who's in the country. It's definitely not reflective of where the burden of disease is, or who are disproportionately affected by these outcomes. And so you see this lack of representation just compounding at every single step, especially as, PGS are often built on the existing literature and the GWAS that we have. And so, back to sort of this conceptual framework, it's a lot, right? It's a lot to go at one point. So it's important to think, how do we start, right? How do we sort of expand our framework from genetics to the trait into a more broader framework of what you're doing here? And so for here, what I'm going to do in this talk is I'm going to walk us through the way that I sort of see it from an epidemiological background of how you build these relationships to get a better idea of what's happening holistically when it comes to the role of genetics and environment in a different trait. And so to look at this, I'm going to illustrate it with using the PAGE study. This is the population architecture using genomics epidemiology. This is a long-running study funded by NHGRI here in the States. And I think it's important to note that this was started by the NIH in 2008 because they already realized that they had a problem with diversity, right? So this is a study that was specifically designed to address the lack of genetic research in sort of non-European ancestries, non-white backgrounds in the United States. And so they brought together a number of studies. This data is the second round of this from 2013 to 2018. With about roughly 50,000 individuals. Now, I know this is not a lot of people for, the way the field is currently, but it's important to remember that in 2013, this was a lot of people, right? So this is sort of what we're looking at. And the people and the participants in this study, they identify themselves as about 22,000 identify themselves as Hispanic Latino, about 17,000 as African American. Then we have smaller groups of people who identify themselves as Native Hawaiian, Native American, Asian, and then the others category. Interestingly, the others category has a sizable number of individuals who are actually of South Asian ancestry, but they did not feel like the Asian label fit for their backgrounds. They put themselves into the others. And so sort of interesting dynamics when we think about how people self-identify within the context of sort of the social political landscape there. Okay, and so what are we going to do? So we have these individuals, and then, there is a wealth of information out there that has shown that we know that these polygenic scores, these tools that collect effect sizes from the entire genome and pull them together, that they were, that they perform differently across groups. There's a lack of transferability across those groups, right? And so I'm not going to recapitulate that for what we're doing today. Instead, we're going to focus on just what happens within a group. And so we're going to focus on the Hispanic Latino PAGE participants for this group. So a lot of diverse within one group. When we think about recently admixed groups and ancestry, and then we're going to further explore what it means when we think about how this is complicated by heterogeneity in environment, right? And by environment, I mean basically everything that's not genetics. That's an obnoxious way to put it, but as a geneticist, it's sort of, how it goes. And so what we're going to do is look at Hispanic Latino PAGE participants, and the trait of interest I'm going to look at is body mass index. Now, I want to say off the bat, this is not necessarily a clinically relevant trait. It's not really sort of the focus of many polygenic score development efforts, but I think it plays a really good role in illustrating a trait that both has genetic components and environmental components with a heritability estimates of around 40%. Okay, so we'll keep going. All right, so where we're going. So this is where we'll get to at the end. At the end, we'll have this sort of idea of how a polygenic score relates to BMI, and then how all these different aspects sort of relate to it. But first, we're going to start with the idea of the population genetics behind it, right? Understanding what the genetic background is, sort of agnostic to outcome within our study group. All right, so here is a PCA. I'm sure many of you have seen these principal components analyses many, many times. And what we have here is that each of these dots is a person, and they are colored as how they identify themselves, right? So we did not do any sort of recoding. We did not do any sort of looking at the data. It's how they self-identify with these racial ethnic labels. And so to orient yourself, on the right, we have sort of the Africa versus not Africa sort of gradient for principal components. At the top, it's East Asia being pulled out towards the top there. Towards the back and left, you can't sort of really see it very well, but it's going to be the Americas, so Amerindigenous ancestry from the Americas. And at the bottom, we have Europe for a variety of reasons. And you can see, if you look on the right-hand side, this is PC 1 through 5, that there is a lot of, continuous gradients across all of these different axes when we look at diversity. And when we got this data back in, I think 2016 was probably when we got the data, it was standard practice to stratify and then do a meta-analysis. And so we look at the data and we're saying, okay, these are the groups. What we would do is stratify by pretty much the racial ethnic group, look for some ancestry groups. And what became really apparent very early on is that there is no sort of distinct way to cluster these individuals, right? There is no line that you can draw and say, on this side, it's this group. On the other side, it's a different group. There's no discrete groupings, right, with these groups, primarily because a lot of them are recently admixed, but also just the nature of human diversity in terms of who's included here. And so it became very important for us to see that this diversity occurred. And so just keep that in mind that the dark blue dots here are the individuals who identify as Hispanic Latino. And what you can see sort of in the background is that they're sort of all over this PC1, PC2 space, because again, of this very diverse, recently admixed background within a group that we often lump together and say they're all the same, which becomes abundantly clear from a genetic standpoint is not the case. Okay, so let's build out this relationship. So the first relationship we're going to look at is how group membership, so group membership that's more granular than Hispanic Latino, can relate to ancestry, right? I'm not going to show you the ancestry to SNP thing, we just showed that with the PCA. So we're going to back up to sort of Hispanic Latino groupings and ancestry. And so I think it's important to take a step back again and sort of see when we talk about the genetics of Hispanic Latino groups, what are we actually talking about, right? What is a definition and what does it mean in terms of the genetics? What I'm showing you here again is the people who just identify as Hispanic Latino. Each dot is a person. The dots that are closer together are more closely similar to each other in terms of the genetic background. Those that are further apart are less genetically similar. But what you can do is you can actually, a lot of these participants were able to self-identify further beyond Hispanic Latino into 3 Caribbean groups, so Cuban, Dominican, and Puerto Rican. And then on the continent, it's sort of Mexico, and then Central America was sort of grouped together, and South America was grouped together. What I think it's important to note is that you do have some structure within this group, right? So it's not a random mix of these sort of dots in different colors, but you do see some clumping because of a shared demographic history within these groupings. And so it's important to note that not only is there diversity within the group, but there's also significant substructure when we're looking at these individuals and how they relate to one another. Another way of looking at this is admixture components. So what I'm showing you here is each vertical line is a person, and their vertical line is colored by the proportion of their genome that is genetically similar to these sort of latent ancestry components I have on the right-hand side. And so we're going to focus on Hispanic Latino here on the right-hand side. And what you see is that there is a large amount of diversity across these groups. It's not unexpected given the PCA. Here, what I'm showing you is if we took those genetic backgrounds and we discretized them into different latent components, so essentially trying to model the recent admixture from these groups, we see a lot of diversity of people with a lot of Amerindigenous ancestry, a lot of African, European, sort of these related genetic similarity components here. And what I want to show you belowhand is that if we further split people up as to how they self-identify, you see differences. Now, I want to be clear, if I picked one person from this, I could not tell you how they self-identify from their admixture proportions, right? I could not tell you if they identify as being Mexican or Puerto Rican or where that might come from. But there are differences on average that can be informative to understanding how ancestry influences how our genetic tools work, right? And so it's important to know, again, that there is an extensive amount of diversity, but also substructure that has relevance to how these individuals move in society in general. Okay. So we've done that, sort of a historical background of that and sort of why, I'm not going to get into why, but there is some substructure of sort of who has sort of different backgrounds and how much and sort of what that differences look like, right? And so now we're going to rely on other literature, so non-genetic literature, to establish relationship between the group membership and BMI. Remember, to be a confounder, it has to be associated with the exposure, in this case, sort of the genetic background, as well as the outcome, in this case, by a BMI. And so we're going to establish this using other data. And so here, we can rely on the wealth of information and wealth of data that have been done, particularly in this particular study that's actually a member of PAGE, the Hispanic Community Health Study and Study of Latinos in the United States. This is a longitudinal study over 4 different recruitment sort of areas in the United States, looking at the health of these individuals. And so what they found in 2012 was that there's differences between these different group memberships in terms of their average BMI, as well as weight gain trajectories. So over sort of age, as well as in terms of how long they've been in the States, if they are sort of first generation. And it's also important to notice that there are differences in the burden of cardiovascular disease risk factors between these groups. Again, so a different relationship and different prevalence and burden of these sort of risk factors and comorbidities when it comes to cardiovascular disease between the groups for both men and women. So that's important. And so we can rely on the non-genetic data to establish this relationship, to make sure that we see, is Hispanic Latino group membership a possible confounder for the relationship between genetics and the trait? And so we've done that. And so now we want to know, okay, we established it has a relationship with the genetic background, it has a relationship with the outcome, but how does it affect the relationship between the SNP and BMI, right? And so for us here, we're asking what happens when this is compounded in a polygenic score? And so this to do, what we did was we applied a published risk score that was developed in mostly European ancestry backgrounds that was published in 2019 to PAGE, and then saw how this risk score performs. All right. And so I want to show you here, this is, we're going to sort of jump into the performance, right? And so this is what I'm showing you is an adjusted R squared for each one of these different groups, how people identify. And I'm looking at the total there includes a full model. So the full model also, in addition to the polygenic score, also includes age, sex, and the study. So the studies are 4 different studies. One is Women's Health Initiative in the United States, the multi-ethnic cohort, which is a study of cancer over the lifespan, and then age data Seoul, so that's what I just detailed, and then one Metropolitan Biobank, BioMe in New York City. And so what you can see off the bat is that the performance of the polygenic score in the darker color differs between groups. And so we can get a better idea of, there's differences between these groups. What you can see here is that if we realign to sort of where the different group members are, you see that the base model, which is age, sex, and study, explains a different amount of variation in these different groups, right? And so you see that in some groups, like in the Puerto Rican individuals here, that age, sex, and study actually are quite informative when it comes to understanding the variance of BMI in the population here. And some it's very little. So in the Mexican-American participants, you can see that age, sex, and study did not explain a lot of the variation. But then we can also petition sort of incremental R squared, right? The partial R squared, that's just the polygenic score itself. And what you can see is that the genetic tool here, the genetic tool to estimate risk, also performs differently in different groups, right? And so you can see that it performs the best in sort of the South American group, followed by the Cuban group and Puerto Rican group, and then it does very poorly in other groups. And so particularly the Dominican individuals within PAGE. And so it's important to note here that, again, it's not that the overall model is doing poorly in that group, it's that these different components, right? So the risk score can do really well in one group, but maybe the other base model is not, which just goes to show the heterogeneity present in terms of how much both these genetic and non-genetic factors matter when we think about how much of variance of BMI is being explained by the model and the variables. Okay. And so, I think it's important to note as, it's very tempting just to say, okay, put it in the model, right? We'll just adjust for it. We can adjust for more and more and more things, and we'll get bigger and bigger and bigger. And as long as we explain more of the variation, it's fine, right? And the problem is that actually, if you do just add group membership as an additional term into the model does worse. And why is this? And this is because you're actually adjusting out a real relationship between outcome and the risk score distributions being different. So what I'm showing you here on the top left-hand side here is the proportion of that population that's in those quintiles for the polygenic score. And what you can see is that the Puerto Rican individuals have overrepresentation in the top quintile of the genetic score, right? So there should be, you'd expect that they were evenly distributed, that, a fifth of the individuals who identify as Puerto Rican would be in the top quintile. And instead, they're overrepresented in that group. And you can see that below in terms of the distribution of the polygenic score is being shifted compared to the others. You see the green sort of moving to the right-hand side there. And so what we notice is that, and I didn't show it here, is that this is actually related to their BMI distribution also being shifted to the right. And so, we're not saying that's automatically all because of genetics, but it is a correlation that we see. And so if we look here on the right-hand side, the difference of that effect size of the beta for the top quintile of polygenic score versus everybody else, and where that is more informative versus less, what you can see here is that for the Puerto Rican individuals, you see this big jump in terms of the risk score, in terms of the beta, right, between that. And what that means is that you're picking up more extreme phenotypes in that group compared to the others, right? Before, maybe you had more of a comparable risk regression coefficient across the different groups. You're picking up, that relative risk comparable between the different groups before adjustment. But after adjustment, what you're seeing is that you're picking up different relative risk across the group, right? For some of them, it's sort of right, well, it's the same. For some of them, what you're seeing is that in order to be in that top quintile, you have a much higher phenotype. And so you're seeing sort of this inequitable distribution of who gets categorized and how sort of extreme of a phenotype they have to be in that risk category versus not, right? So you're not really actually helping yourself here. And especially if you're thinking of more of an equitable framework, you might be exacerbating these difficulties when we think about prediction between different groups by adjusting, or by forcing everyone to have the same distribution. And so depending on, again, your goal for this, we really don't want to see that. Okay. And so where we're going, right? So we wanted to expand out to that. And I'm going to go on this part to more of the biomarkers and social and structural determinants of health aspect. And so in order to do that, I want to take one sort of step into the background of what we mean by environmental context. And so there are a lot of different frameworks we could use, but I think the most important thing for us to note is that it varies greatly in resolution and generalizability, right? And so here on the left-hand side, we have this sort of really micro scale measurements. We think about genetic variants and then even multi-omics like methylation. And as we get larger and larger in scale to the individual, such as biomarkers and lifestyle, interpersonal relationships, neighborhood relationships, societal relationships, you see the scale getting bigger and bigger and from the individual level to the neighborhood level, all right? And so when we think about this, we have to really consider the study design to get at this because, again, you have significant substructure and the meaning of these variables changes depending on their context. And so we're going to look at that here in terms of what that means for our polygenic score. So we're going to first look at the biomarkers or individual level risk factors sort of in the pink box here, and then we're going to go to the more macro scale neighborhood factors when it comes to social determinants of health in the sort of green-blue after that. And so we can sort of idea for an individual factor, we're going to look at smoking, right? So smoking is an individual, risk factor for different things. And one thing that we can examine this is say, okay, we know that a GWAS is essentially a bunch of correlations, right? And then if a polygenic score is built from the GWAS, it's basically summarizing a ton of correlations, right? And so we want to know what else it's picking up, right? Because the study population where it was developed might have other factors, other confounders it's picking up. So how does it affect us? So what I want to show you here is when we flip it on its head. So we have the polygenic score, and we're seeing how much other components explain the variation in the polygenic score itself, right? So the idea is, what is the polygenic score actually capturing? Is it capturing trait differences? Is it capturing ancestry differences, other things? And we can check. And so what we see overall within Hispanic Latino individuals on the left-hand side here is that, about 18% of the variation in the polygenic score is explained by this amerindigenous admixture component within these groups. And only 3% is explained by BMI, right? Which is equivalent to the sort of the adjusted R-squared or incremental R-squared of about 3% when we looked at the models of 4. But it's important to note, again, because of differences in genetic background due to genetic drift and different forces, ancestry matters a lot. And so we can look at the more granular groupings here. I'm showing you the Mexican versus Puerto Rican because it's the largest groups on the page. And what you can see here is that the amerindigenous ancestry matters more for the Mexican Americans than it does for Puerto Ricans, mainly because there's more of that ancestry in that group, but also the variation present was captured with those risk scores and relatively comparable amounts of BMI being captured. But what I want to show you on the right-hand side here is if you stratify by smoking. So smoking is a lifestyle factor, right? It should not affect the genetic variants, and it should not affect really, how those are captured. But what you can see is if you stratify by smoking, right, it doesn't really matter for Mexican American individuals, right? The AME proportion, the same amount of the polygenic score is explained by variance, by ancestry, same amount for BMI, right? Relatively same. but it's very different for the Puerto Rican individuals. And so you see that ancestry matters much more for those who have ever smoked versus not ever smoked. And so here you see that there's sort of this interaction and sort of this sort of intersection between a lifestyle factor, how much ancestry matters, and sort of how you would need to adjust for it when it comes to this polygenic score that's based off of these sort of millions of correlations, right? And so it's important for us to know. And again, it really depends on the prevalence of these variables in the training versus test set. It's important for this sort of population level characterization. And so we see here that sort of group membership might affect smoking and then traits, but we're going to expand out and look at social and structural determinants of health, right? So more of a macro scale look. So social and structural determinants of health, this is by the CDC. They categorize it as roughly 5 different categories, looking at education, healthcare, the neighborhood and built environment, the social and community context, as well as economic stability, right? So these are large sort of categories, that umbrella categories we can think about as to how the health of individuals in populations can come to be. And so we are going to look and try to build these relationships again. And so in order for this to matter, again, it has to be related to both the exposure, in our case, genetics, as well as the outcome, in our case, BMI. And so we can look at literature for this. This is sort of looking at an older 23andMe paper, looking for individuals who identify as Hispanic Latino. And what you can see is that across the United States, people who identify as the same identity have different genetic backgrounds, right? In terms of average ancestry components. And so you see, and this makes a lot of sense given the history of these groups within the United States and the history of the United States in general. But you see sort of, again, the highest indigenous ancestry along the border with Mexico, which is explained, by history there. And then you see the differences, right? And so we have a substructure that relates to geography within a group, right? So it's important to think within one group, how people identify means different things in terms of what the genetic correlates might be, depending on geography. And we can look again at the sort of outcome measure. And so this is work done by other folks in my department, looking at structural racism and BMI of the United States. And they had a county level measurement model for this, a metric, looking at metrics across multiple of those domains for these social and structural determinants of health. And so here the focus on sort of black-white differences in the United States, but you can imagine they would extend somewhat to the Hispanic Latino groups. And so what you can see again is those differences in terms of the structural racism index between these different geographic counties within the United States. And that, what they found as well is that is a higher county structural racism associated with higher BMI in Black individuals compared to white individuals. This was after adjusting for what you'd expect, sort of county income, when they found that, again, more money equals lower BMI, according to the associations. And so really just to bring it back together, saying that there's differences for us in ancestry and geography. There's differences in terms of the social and structural determinants of health and how they relate to the outcome by geography. And so let's see if we can bring it together. And so one sort of regulative domain that we're really interested in within this group and population is sort of the idea of immigration, right? So how does immigration take place and sort of why does it take place and what does that have the effects of the health in these groups in first, second, and third generation of individuals here in the United States? And so we can look at these studies here. And what they found is that one of the strongest predictors of moderate and extreme obesity in these participants was a length of residency in mainland US. This was actually consistent between different background groups, but it was interesting that it's not acculturation that's associated with obesity, but rather just how long you're in the United States, which they consider an obesogenic environment. Okay, so again, we're just pulling together these threads to show sort of how all these things matter, how the lack of, the immigration would matter, the social structural determinants of health would matter, relying on this data to pull it together, right? And so we can do this in one big model. This is work that's been led by Lindsay Fernandez-Rhodes and Kristen McCardle, her then postdoc, looking to see is there a relationship and interaction between age of immigration and a polygenic score for BMI. So they had a much expanded model, including education and income. But the idea that I want to point out here is that if they look at this interaction term where they're modeling how does the age that someone immigrated to the United States, how does that sort of modify how strong or how good of a tool this polygenic score is to predict BMI? And the reference group here, again, is over the age of 20. So if they immigrate to the United States over the age of 20, what you see here is that those individuals who immigrated to the United States between the ages of, when they were born to 5 years of age, actually their risk scores were better performing than others, right? They have this positive effect size, meaning that the effect size of the progenitor score is also, stronger. And that it's interesting in that it was actually stronger in women than it was in men. So here you see there was this factor of when people have come to the United States, again, adjusted for age at this point, but when they came, at what age and development you can think of, they came to the United States, can alter how well a genetic tool does in that population, right? So again, that E modifying how G predicts the outcome. And we can look at group membership here, right? And so what we can see here is that if we look through here, that again, the model fit differs by background. This is sort of similar to what we saw before. The numbers are higher because they have much more terms in their model, but sort of similar trends. And we can see again, the effect of the PRS itself differs between these different groups with much stronger effect sizes, in this case, in sort of the Puerto Rican individuals compared to other groups, like the Mexican individuals here. But what I think is really important for us to note is that the interaction of immigration and PRS on BMI also differs by group. And so what you can see here is that the effect size of age of migration of those when they're very small versus over the age of 20 is much, much stronger in the Cuban population. So those who came as small children, the risk score is really a lot better in them than people who came later. While it's actually the opposite for those from South America, in which those who came as small children actually do much worse, right? And this is important because, coming from the world of genetics, a SNP is a SNP is a SNP, right? It doesn't matter sort of how you measure it, a SNP is what it is. When it comes to when we expand ourselves, look at these environmental variables and social determinants of health, it's a simple measure, right? At what age did you immigrate to the United States? But it means different things in different contexts because people come to the United States for different reasons, under different sort of time periods, to different areas in the United States in terms of where they settle. And so it's important for us to think about, again, the context is what matters. And this is all very, very complicated, right? It gets very, very complicated very, very fast. But I think it's important for us to take into context that these things do matter, right? We kind of can't stick to considering just putting as much genetic data as possible and expecting that resulting risk score to work for everybody, right? Because as we move again from discovery to this outcome, things change. So I'm going to hurry up because I know I don't have a ton of time. I need to sort of finish wrap up these slides. But a lot of things that I work on right now, again, is defining these populations, these study groups that are relevant to a specific study question, right? And so this is a lovely quote that is, to group is natural, but there's no natural groupings, right? And so how do we think about how we group individuals in genetics research? And a lot of it is a sort of lack of precision when we talk about groups. So it's very difficult to assess accountability or assess equity if we don't actually know what we're measuring, which is a sort of a concern here. And again, from an epi standpoint, we don't really know how our study groups relate to target groups unless we have better precision of how we describe individuals. And you can see this in terms of when we look at the literature and the databases that have curated our sort of wealth of knowledge about the role of genetics in these complex traits. And here, looking at the GWAS catalog again, I'm using European as an example, but it's important to note, in order to do this, in order to have this amount of accessibility, looking to see what's there, and the accountability that we want to have, that we make sure we're doing it in diverse groups, they have to collapse concepts of genetic ancestry, of geography, of nationality, of race, which are not sort of a one-to-one relationship. They're not directly related, but it had sort of happened. This imprecision had to happen because there's a lack of information, there's a lack of standards, a lack of precision when it comes to how we describe our study populations. And often this comes from a fundamental tension, right? It comes from a fundamental tension between the technical motivations of how we group individuals, which is maybe sort of the assumption of homogeneity in the group, and the societal context in which people actually live, right, and function and interact with the world in terms of race, ethnicity. And these are not the same thing. They're not the same thing. They might be correlated in some contexts, but they are not the same fundamental construct, right? And so again, you have ancestry on one side, and you have race, ethnicity on the other side. And again, it's sort of, you can't just use one for the other and accept, the other way around. And this sort of fundamental tension we sort of deal with on, a regular basis in genetics research of how you define the groups, how do you use the results, how do you interpret the results, because of, again, this conflict between the two constructs. And so I was lucky to be part of this National Academies report that came out last, I guess, 2023, spring 2023, and looking at how we use population descriptors, right? And so one of the main points about this is having more precision as to how we describe groupings, to look at genetic similarity and sort of how we would model that. So, I think part of this is that if we are looking at genetic similarity, so how similar people are depending on their genetics, then we should group by genetic similarity. It doesn't really make any sense to group by racial and ethnic lines. And I showed before, particularly in these admixed groups, right? So it requires us to really question, is what we're doing what we say we're doing, right? When we say we have the genetic ancestry groups, but we're looking at just racial ethnic groups, do we actually have that level of exchangeability within the group? I would say not. And this is particularly in the sort of the face of this large push towards data-driven work, right? And I think it's important to say it's data-driven, but where exactly is it driving us to, right? And so it's important to take a step back and see, okay, where is this data coming from? It's important to know the system itself, which it comes from, is faulty, and which a lot of times these algorithms actually rely on reference data, which is labeled as per social constructs, right? So you're not actually getting away from the social construct part of it by using the reference data and these machine learning algorithms when it comes to clustering, but instead, you're just obscuring sort of what's going on there, right? So it's important to be very intentional, and this can be done. It has been done well in some instances, but important to take a step back that sort of, the computers won't save us, right? I don't think anybody ever thought they would, but they really can't. And this goes for how we cluster individuals, right? And so what I'm showing you here is a real section from a paper where they clustered individuals based on their ancestry. And I think in the bold where I'm showing you that they had a sort of this decision boundaries for things. And so if anybody had a likelihood over 0.3 for more than one group, they arbitrarily assigned these groups to this sort of hierarchy, right? So African over European, Hispanic over European, Hispanic over East Asian, et cetera, et cetera. And what I think is important is that they put the arbitrarily in there, but really you put these decisions into it. And what you're doing is you're bringing biases and the way that people think about the world into the genetic space, right? And so if we look at the sociology of how we think of groups, we can actually draw directly upon this. There's this one theory here about the two axes of subordination, looking at sort of distalness from whiteness in terms of these different axes of inferior, superior, and foreign American. And what you can see here is, again, it's a sociological sort of theory. I'm sure people were not developing these sort of clustering algorithms thinking of this, but it does recapitulate them, right, in a way that is beyond the one drop rule, beyond these rules of hyperdescent, but really these different axes of how we consider individuals. And then if we think about, if we're trying it from a genetic standpoint, right, and you're saying you want to have a certain level of homogeneity, it makes even less sense, right? Because you have these groups that if they have any bit of that group, that rules of hypodescent, the one drop rule, you're putting them to that group. But if they're already the most diverse group there is, what are you actually making homogeneous here? Right? What are you actually ensuring there? And so it doesn't make sense in terms of an environmental context. It doesn't make sense in terms of a genetic context. So how can we do better? So this is sort of one thing that we're sort of ongoing work within my group right now about how we can actually do better. This is the idea of looking at genetic similarities, specifically for polygenic risk scores. The idea that right now we tend to group people and give them their risk back based on these sort of genetic ancestry groups that are often just recapitulating racial categories. And so instead, the idea is if you want to have that level of exchangeability and the comparison between different groups, between individuals within one group, then you should group people by their nearest neighbors, right? How close they are genetically speaking. And if we want to have precision health and precision medicine, that includes precision as to who you're comparing people to, right? And so for us, what we're saying is each person can get their own reference population and then have that new individualized group to have a better idea of relative risk with people who are more directly comparable to them because they're genetically close. So let me just show you what sort of that means in terms of the concepts. And so here we're going to have an individual who is identified as Hispanic Latino. You can see them in the white dot there with the black sort of circle around it. And what you can see is that their nearest neighbors are sort of clustered in this area. people are categorized by how they self-identify in this group. So what you can see off the bat is that they're not all the same, right? People's nearest neighbors are not always people who identify themselves as the exact same racial ethnic background, which is what you expect given shared ancestry between recently admixed groups. And so, this, again, it doesn't really matter in the center, right, when we think about the means, but it really matters when we look at the tails of these distributions. And so what I'm showing you here is PC1 and PC2, PC1 and PC3. Each person is an individual. They identify as Hispanic Latino. And I'm showing you what proportion of their nearest neighbors are also identifying as Hispanic Latino. And what you can see is if you go towards the edges, right, these outliers who are Hispanic and or Latino, that is our identity, it's not incorrect. You see that their nearest neighbors are not in that group. And so if you were comparing them to everyone else, you'd be doing them a disservice because they're not actually directly comparable when it comes to the genetic background. And you can see this again, another way of looking at it in terms of the concordance on the y-axis and the PC1 on the x-axis. And you can see this both for Hispanic Latino individuals and African American participants in PAGE, where again, if you look sort of at where the majority of people are, yeah, their nearest neighbors are the same background as them. But if you look further and look at the tails, you see a drop-off in terms of where their nearest neighbors are. Again, showing that people's closest genetic sort of backgrounds and most similarity is not always within a racial ethnic group because a racial ethnic group does not symbolize anything discrete when it comes to genetics, right? It does not represent these genetic ancestry groups. They are sort of different constructs that need to be considered as such. And we can look at that for individual reference populations. So here I'm showing you 4 different individuals from this study group. And so A, the person identifies as Hispanic Latino, and 996 out of 1,000 of their nearest neighbors also identify as Hispanic Latino, with 2 people who identify as African American or African and 2 as other. And you see sort of a similar dynamic with B, sort of a different area where now you actually have a small subset that actually identify as Native American as the reference group. On the bottom left-hand side, you have individuals who actually the majority of their nearest neighbors are African or African American because of the amount of African ancestry they have in their background. Again, not surprising because of the diversity within this group. And on the right-hand side, I want to point out that this is an individual whose actually nearest neighbors are Asian or Native Hawaiian, right? So you see this large group where their nearest neighbors are not Hispanic Latino. And this could be one of 2 things. It could be that they are Hispanic Latino, because again, it's a group defined by coming from a country that speaks a certain language, right? It does not come from having any sort of genealogical certain generations. Like it's not that kind of a label. So it could be that, and those labels are complicated. The histories of these areas are complicated. It could also be that they were misclassified somewhere along the way, right? Where they have a Spanish last name. In this case, maybe they're Filipino because of the history of colonialism and empire in terms of the Spanish. And so we don't know. But again, it goes to show that not only are the nearest neighbors not necessarily in the same ethnic group, but also the labels themselves are also subject to interpretation differences in terms of where they're collected. And so they're not as sort of infallible as we think they are. And so we can see again that here, this concordance that goes down. And the idea that, again, these admixed groups have shared ancestry across them, right? There is no discrete groupings that you can say on this side, it's this group, on the other side, it's another group, right? That's not how it works. And so, to sum it up, really, I'm going to sort of finish up here and if we get to questions, I understand that the point of this talk, I keep on telling you that maybe we tend to have better granularity, right? Better precision, so smaller and smaller data sets. But there is a cost to this, obviously. The sample size is smaller. You're already starting with a smaller pool of people. And I'm essentially asking you to make it smaller and smaller and smaller. And so there has to be some sort of balance that we approach when we think about our study design, right? Like what can we sacrifice in terms of sample size to get precision of question and maybe a stronger and better, more valid effect estimate. And these are all conversations that have to happen at the study design part, right? At the analytical plan development. It has to happen at every single stage. It can't just be the default of putting everyone together, right? It has to have thoughtful ideas put behind this, which again, I don't think is novel. It's sort of a novel thing to put forward to people, but I do think it's important to sort of bring it up and put that sort of epi back into the sphere. And when we think about the epi, again, this trade-off between power and precision, I think it's important to note that there is no study we have that is large enough that you don't have to worry about bias, right? I know the idea is that bigger is better, but bigger study, bigger bias, right? It just makes the thing there. And so you have to have precision in your question. Otherwise, you may not have power to detect it anyway, right? Okay, so what does it mean for genetics in general? I think it's important to note that what I'm trying to show you here is that we just have to think critically about who we include and who we model as a distinct population, right, with our studies, what we think that's saying in terms of the genetic background and the lack of, or the extent of homogeneity there, what that says about the environmental and epidemiological background and perspective as what we think is all the same, and to not sort of flatten that. The other thing is that admixed groups and individuals are not just some of their parts, right? They are individuals that exist in communities and societies. And as a geneticist, it's sort of not my job to sort of see them as like, oh, well, they also have African haplotypes. They also have European haplotypes. It's yes, but that doesn't mean, again, they're some of the parts, and everything's very complex as we move, especially towards this precision health sort of idea. And this is going to require us to have this reconciliation of how do we model the interface of race, ethnicity as a social construct, as genetic similarity when we think about the genomic substrate. This is an older framework from 2006. And I think as we move to more of these multi-omic approaches, multi-stream data, sort of appropriate considerations, this is going to become more and more of a salient thing we have to think about of how do we reconcile environmental context, which is more related to race and ethnicity, with the genetic components we're trying to model, which really is about genetic similarity and how do they interact with each other. I just want to say, I don't think we all have to be experts at everything. We should not be experts at everything, right? This sort of work requires deep and trusting cross-disciplinary collaboration. When it comes to the acculturations that I showed you with the age of migration, Lindsay Fernandez-Rhodes is trained in social epidemiology and has been a great collaborator to sort of get these different perspectives for. And I think we need just to think about the context, right? And sort of think about how we work together, maybe outside of what we think is our immediate realm, especially as we get sort of wider and wider data sets to better have modeling. And this includes, questioning the assumptions and the defaults that we have, right? There's a difference between assumptions that we make for the modeling itself, the statistical assumptions, and what we accept as the default, right? And I think we cannot get to equity until we consider what the default is and sort of try to break out of that sort of black box of what that means, right? Including the questions we ask, who we include, and the systems that we model. And so the last point I want to make is that I think it's important to take a step back as well, and I keep on saying that phrase a lot, but I think I mean it in terms of what systems are in place and how can we change them as a field, right? And so we have this GWAS catalog, and I'm showing you the proportion of the GWAS catalog participants that are sort of grouped these rough categories, right? And they do not represent the world population, the global population. They do not represent the US population coming from a US sort of perspective, but they do represent the investigator, the scientists, us, right? And I did not go into this for purely selfish reasons. I'm sure you didn't either. And so I think it's important to think, okay, we want to tackle these issues, but we also have to have a certain level of cultural humility about diversity of who was in our workforce as well, to sort of see, where are areas that we can work better on, how can diverse perspectives be integrated to do better work, and so we can have maybe a better path forward when thinking about health equity, including genetics and genomics. And with that, I want to say thank you to my lab and to people that I worked with for collaborators and Paige and Lindsay. And I'm happy to take any questions people have. Thank you.
We have one hand up. Hello. Do you want to unmute and ask your question?
Yes. Hi. So
Hi.
can you hear me well?
Yeah.
Mm-hmm.
Okay. Hi. Thank you so much for the talk, Professor Wojcik. I love all your research as always. I'm very excited to see publications addressing these issues so important, and you're doing it so well. So I have a question, is more like open-ended questions. So with
Yeah.
these PRS, this new diversity, like genetic diversity measure,
Mhm.
what would the idea be moving forward for GWAS or PRSs? Would it be, I know you said not just to account for other type of diversity, and I agree. I think it should be like very trait specific. But how would you see them related to the environment? Or like how could we actually set up a framework for researchers for when they're doing GWAS or when they're gonna calculate PRSs and how to, yeah.
Yeah, that's a great question. I mean, I think it's sort of a big question, and I have sort of a couple of answers for it. So one, I think when it comes to GWAS, I think the goal of a GWAS is fundamentally different than a polygenic score, right? The goal of a GWAS is theoretically discovery, right? And so I think in that front, I come from an epidemiology background and we're always shouting about study design. And I especially come from a background looking at infectious disease, right, in terms of our genetics. And so one of the main things I think about is having, again, if you're making a comparison of association, having the relevant groups at present. So for example, for infectious disease is that in order to have an infectious disease, you have to be exposed to the pathogen in the first place, right? And so when it comes to looking at these larger GWAS and what this, you saw this with COVID, right, where the different GWAS, the results changed longitudinally as you had different populations being infected and being sick from it. That again, there needs to be a granularity and a sort of a clarity of question where you're comparing your cases to actual controls, right? And for some things, the use of population controls doesn't matter as much. Some things it really does. You're picking up other things of why people are getting sick versus not. And so when it comes to GWAS, I think there's some benefit to having, again, clarity of that question when it comes to context, right? So in that case, you're more like subsetting into context and figuring out in which context are you actually trying to get that comparison to be as clean as possible, where you're looking at the only difference would be a genetic marker, right, to get that effect, strong effect size. When it comes to polygenic scores, I think there's a variety of things you could do for that. One of them is that, a lot of times right now for polygenic scores, you go into the clinic, a person will go into the clinic, and they individually will be assigned to a group. And then your relative risk or absolute risk is given back to you based on that group, whether it is sort of European ancestry, roughly whatever it is, right? And I don't think that's actually the proper way forward because you have two problems. One is with the admix group I showed you before, where you have increased variation. The other is that you have people who don't fit in a group, right? They're never going to fit in a single group. They exist between those boundaries and they sort of fall through the gaps, right? And so if we're going to have polygenic scores be developed, I think we need to adopt these sort of principles of inclusive design to have it so that anybody who walks through the door would have some estimate given back to them. Again, because here you're looking at saying, why do we even have to have those groups, right? Are they informative of what we're doing in the first place? And so that's sort of a different question and a different sort of framework to think through than a GWAS where you're thinking about discovery, right? Because here you're thinking again, looking at relative risk at a population scale, looking at those different factors. And then additionally, I think, I mean, I say this kind of shooting myself in the foot for this, but not everything is because of genetics, right? And so I think the idea is with context and polygenic scores, some things don't need a polygenic score, right? Sometimes a polygenic score is not actually that informative and it doesn't need one. And I think defining context in that front, both in terms of the populations, the genetic background, racial ethnic background, the environmental background, is important for us moving forward because it's sort of, I think we'd love to say that we have a genetic code. It's like, Gattaca, and then you know exactly the future, but that's not how the world works. And I think that we can only do better by acknowledging that. A very long-winded answer to that question, but yes, thank you.
No, thank you.
More questions from the audience? Let me ask a question. So I guess part of what I'm hearing is, well, like, do we really need groups? Like,
Yeah.
can we use multivariate measures, instead of groups?
Yeah, I mean, exactly. I think this is this already happened in the GWAS space, right? Where before you would have to sort of stratify and then do a meta-analysis. And now there have been more, recent methods that can directly account for relatedness in a way that you can have a meta-analysis. You pull everyone together, you don't need to sort of stratify. And I think, it's sort of maybe not the same exact sort of idea with PRS, because again, you don't want to sort of just pull everyone together and just call it a day. But the idea of just questioning, again, does this standard practice have to happen that way? Or can we develop novel methods that get to the heart of the problem or heart of the issue at hand, that don't require what we think of as, a standard practice, but it's no longer needs to be a standard practice because methods have changed, data has changed, our data sets have changed. And so, yeah, just questioning whether we have to group at all. And maybe sometimes you do, but I think not all the time. And those cases we could do better.
Hmm. Other questions? I guess I have one more question at least. So, I mean, when people have been thinking about sort of transferability of, polygenic risk scores,
Yeah.
You can kind of try and divide, partition it into, okay, there's the effect size part and there's the weight part and
Yeah.
maybe the allele frequency part. I mean, do you think there's going to be some clarity on that? Um, you know, is it
I
really
mean,
good that the effect sizes are more transferable, for example, than say the allele frequency or the LD part?
yeah, I mean, I think unfortunately, I think there's no answer for every trait. It's very trait specific. And so I didn't show these results, but it sort of depends on the trait itself and its relationship with ancestry, relationship with demographic histories. Additionally, I think there's sort of the genetic under the umbrella of genetic architecture and the things you want to sort of look at and see why you don't have transferability. I think one maybe aspect that's maybe understudied, and there's some groups who have done this really well, is looking at heritability, right? So how much does genetics even matter in the first place, right? When it comes to explaining the variation. And so it could be allele frequency, it could be the LD is not being captured, which is, better and better methods to sort of account for that redundancy with LD. But also, the ceiling is different depending on context. And so is some of the transferability issues because it's less heritable in that context, right? And therefore you would never get to that high. I think that's sort of an unknown when it comes to real world data and identifying where the better context is. We see this somewhat when it comes to, we have some preliminary data looking at hypertension in New York City as a function of air pollution. And what you can see is actually a measure of income level, and that those individuals who have, are more wealthy have access to a lot of interventions, and therefore the only thing left to explain the variation is genetics, right? Not the only thing, but more of it is left for that. While those individuals who are maybe of lower income level, who don't have access to a lot of these interventions, the environmental variance is greater, right? And so therefore the heritability is sort of different as well. And so I think it's all those things, and it depends on the trait, it depends on the context, which I know is not sort of what we really want to hear when a lot of us are developing frameworks that can sort of pipelines put things through. But this is where, again, I think domain expertise really matters, and understanding the trait and interest and collaboration with not only people who specialize in the exposures, right, genetics, sort of the social determinants of health, but also the outcomes and the communities in which we're working with. And so, I mean, I would love if there was one answer in terms of it's always this or this or this, but I think it's just very, very dependent on everything that goes into it.
Michael Wilson, go
Hi,
ahead.
thank you. Thank you for a very interesting talk. One application of polygenic risk scores of late, certainly being discussed, is for risk stratification for breast cancer and mammographic screening. And it
Yeah.
seems that one of the key implications of what you've been telling us is that there's going to be heterogeneity. Now, one of the things about the risk prediction software is that it needs to be approved as a software as a medical device.
Yes.
Any thoughts on how one should be, if you're in the regulatory chair, how you should be dealing with the uncertainty of the predictability of the polygenic risk score if it hasn't been estimated in the really polyvalent, or I don't know what you want to call it,
Yeah.
manifold way that you've been describing?
Yeah. I mean, short answer is no. I have no answers for that. I think it's sort of outside the realm of my expertise for sure. But also, I think there are efforts who are trying to address that, right? How do you deal with the implementation, not only the regulatory aspects of it, but the implementation of this when it comes to that heterogeneity? And I think you sort of run into the risk of there's that tension between, do you approve something for one group knowing it'll do well in that one group and not as well for everyone else? or do you hold it back until you have a more equitable tool, right? And I think we've seen the long running history of any kind of tools in the health clinic that we've favored this idea that for somehow trickle down theory is going to work for these tools as it surely did for economics. And so, you know, I think there's a tension, and it's again nothing I think is really easily solvable. There's efforts within Emerge right now to try to determine sort of how that would work within the health systems they have. And you can sort of see the different approaches they've taken with the ethical sort of qualms about that. But no, I think it's very difficult. And I don't know. And additional with the FDA for us here, and so the regulatory background, there's been some debate as to how you actually define those groups for how well they do differently across the groups, right? What are the relevant groups for that definition? And so we'll see how that works out. There's another national academies here in the States that's coming out with a report at the end of the month as to how to consider race and ethnicity in biomedical research. And so maybe that can give like some sort of insight into it. But I think, you know, a short answer after I rambled for a while is no, I have no idea how that works. Yeah.
Thank you.
Yeah.
Any last questions? Oh, Esteban.
Hello. Very, very fascinating talk. It was really interesting to hear your in the presentation about interactions between the polygenic scores and some interesting things such as, for example, immigration age. So my question is, I mean, this points to the fact that in future, for future risk prediction, it may be important to include in the models. I mean, ideally, the polygenic scores plus socioeconomic scores, but also possibly interaction, no analysis for this. What are your thoughts about this?
Yeah, I think was where it would ultimately need to go to some extent. You sort of see this in the sort of the non-genetic clinical risk tools, and you have sort of interactions with things. I mean, I think it'd be very difficult. I don't think we're there yet, because I think when it comes to the social terms of health and how we measure that and how we sort of develop those scores, that is a whole realm of people sort of spending their careers looking at that in a way that's transferability, right? Because I mean, again, for SNPs, if you have a score, right, if you have an array, you can measure it, and it doesn't really matter where you do it. There's slight differences, but it's relatively reliable estimates. When it comes to social determinants of health, individual measures mean very different things depending on where you are. I think air pollution is a great example of this. Where in New York City, the higher areas of the higher air pollution levels are in areas of greater socioeconomic status, right? In terms of like Manhattan and where you are, where if you go down in air pollution, you have a sort of a decline in that socioeconomic status, which is very different from other metropolitan areas and where there's much more air pollution in socially deprived areas, right? So again, I think it's the complexity is much more. I think we're really sort of just beginning to see how much that would matter. And again, some things it doesn't, you know, what we think might matter for these variables in one group doesn't actually matter in a different group. But I think it's one of those things where in order to know that, we can't rely on these sort of algorithms that, you know, create these different latent variables, to put them together and say, look, I made a big model. You kind of have to think about the context. And this is why you've seen, I think, a lack of transferability with these methods for the environment and everything from UK Biobank to the US, right? Because the social context is very different. The same factor means something very different. And any model transferred in one group is even less transferable to anywhere in the United States than the genetic score would be, right? Because there's more of a apples to apples comparison there. And so, yeah, I think that would be great in the future if you could go in and get your individualized risk based on everything, you know, a more holistic approach. But I don't think we're there yet. And there's a lot of work to be done by a lot of different people that, you know, hopefully we can have opportunities and funding and data to do that. That'd be great.
Okay, I guess we better wrap up for today. Thank you to our speaker for a really fascinating, thought-provoking talk, and we'll look for everybody at next month's International Speaker Seminar. So thank you very much for your attention.
Thank you, everybody.
Bye.